Buvalve mollusk. Some knowledge of bivalve biology is necessary to understand operations of a bivalve hatchery and to assist in solving problems that arise. It is not the intention here to give a detailed description of bivalve biology, but to provide a brief resume of information pertinent to the operation of a hatchery. Bivalves belong to the phylum Mollusca, a group that includes such diverse animals as chitons, chain shells, gastropods, stash shells, or cephalopods, squid and octopus, as well as clams, oyster, mussels, and scallops. The phylum has 6 classes, of which one is Lamellibranchia or Bivalvia. These animals are compressed literally, and the soft body parts are completely or partially enclosed by the shell, which is composed of the two hinge bulbs. The gills or setinidia of animals in this class are well-developed organs specialized for feeding as well as for respiration. External anatomy. The most prominent features of bivalve is the two bulbs of the shell that may or may not be equal and may or may not completely enclose the inner soft parts. They have a variety of shapes and colors depending on species. The bulbs are composed mostly of calcium carbonate and have 3 layers. The inner or nacreous layer, the middle prismatic layer that forms most of the shell, and the out layer or peristachum, a brown leathery layer which is often missing through abrasion or weathering in older animals. And this is the part of bivalve mollusk: hinge, ligament, umbo, hinge teeth, muscle scars, growth lines, and ventral margin, and pallial line, pallial sinus. Bivalves do not have obvious head or tail regions, but anatomical terms used to describe these areas in other animals are applied to them. The umbo or hinge area where the bulbs are joined together is the dorsal part of the animal. The region opposite is the ventral margin. In species with obvious siphons, the foot is the anterior ventral position, and the siphons are in the posterior area. In oyster, the interior area is the hinge. In its scallops, it is where the mouth and rudimentary foot are. And this is Ostrea edulis and Argopecten gibbus. The soft tissue anatomy of the European flat oyster, Ostrea edulis, and the Caligo scallop, Argopecten gibbus, visible following removal of one of the shell bulbs. K. Am adductor muscle. G. Gills. G. O. Gonad. Differentiate as O. Ovary and T. Testis in the calico scallop. L. Ligament. M. Mantle. And U. Umbo. The inhalant and exhalant chambers of the mantle cavity are identified as IC and EC respectively. Mantle. The soft parts are covered by the mantle, which is composed of two thin sheets of tissue thickened at the edges. The two halves of the mantle are attached to the shell from the hinge ventral to the pallial line. Adductor muscle remover from the mantle shows the underlying soft body parts or prominent features of fisher adductor muscles in the marine species or the single muscles in a monomarian species, oysters and scallops. Gills. The prominent gills or Setennida are a major characteristic of Lamabellibranchis. There are large leaf-like organs that are used partly for respiration and partly for filtering food from the water. 2 pairs of gills are located on each side of the body at the interior, and 2 pairs of flaps. Foot. At the base of the cerebral mass is the foot. In the species such as clams, it is well developed that is used to burrow into the substrate and another animal in position, and scallop muscles opening from the basal gland through which the animal secretes are thread-like elastic substance called byssus. digestive system. The large gills filter food from the water and direct it to the labial pulps, which survive the mouth. Food is sorted and passed into the mouth. Bivalves have the ability to select food filtered from the water. Gonadal development and spawning. In most bivalves, sexual maturity is dependent on size rather than age, and size at sexual maturity depends on species and geographic distribution. Production of eggs and sperm is termed gametogenesis, and size of the bivalve along with the temperature and quantity and quality of food are undoubtedly important in initiating this process. Several methods have been used to determine if a bulb is ripe and ready to spawn. The most accurate method is to make histological size of the gonad. Bivalves that reach the size of sexual maturity and spawn for the first time are sometimes referred to as virgins. Although these animals attain sexual maturity, the number of gametes produced is limited and sometimes not all are viable. The period of spawning in natural populations differs with species and geographic location. Spawning may be triggered by several environmental factors, including temperature, chemical, and physical stimuli, water currents, or a combination of these and other factors. Embryonic and larval development. These topics are covered more fully in later sections, but a brief description is given for continuity. Eggs undergo meiotic division at fertilization to reduce the number of chromosomes to a haploid number before the male and female chronologically can fuse to form the zygote. Two polar bodies are released during meiotic divisions, and when apparent indicates successful fertilization, cell division begins, and within 30 minutes for fertilization, the eggs divide into two-celled stage. This is embryonic and larval development. Spawning adults, next fertilization, and the larva stage, mature larvae, early juvenile, and grow out. When larvae approach maturity of food developments and gill, the rudiments become evident. Small dark circles, dots, that eye spots develop near the center of each bulb of some species. Between periods of swimming activity, larvae settle and use the foot to crawl on a substrate. Metamorphosis. Metamorphosis is a critical time in the development of bivalves during with the animal changes for swimming, planktonic, and sedentary benthic existence. Considerable mortalities can occur at this time, both in nature and in hatcheries. The subject is dealt with in detail later since the important aspect the hatchery production of juvenile bivalves. Growth. In general, statements can be about growth in juveniles and adults since it varies greatly between species, geographic distribution, climate location, and subdital or intertechnical zones. Growth can be measured in bivalves by different methods including increments in shell length or height, increases in total or soft body weight, or a combination of all these factors. It virtually ceases in winter, resulting in annual checks in the shell. These winter checks can be used to age some bivalves. Some species are short-lived, but others may live to over 150 years.